In this video, we are going to continue talking about the structure of nucleic acids. So as a review, when it comes to DNA and RNA, understand that there are different components to each of these nucleic acids. Understand that DNA is going to have the bases adenine, guanine, cytosine, and thymine. Right here, what you can observe is the one letter code for the nitrogen bases. In the case of RNA, we're going to have adenine, guanine, cytosine. It's just that now we have uracil. Remember that the sugar structure that is found in DNA is deoxyribose, while in RNA it is ribose. When we're forming nucleosides and nucleotides, remember that the nitrogen base are the ones that are outlined in the first row. We are going to have a deoxyribose sugar motif in DNA while a ribose motif in RNA. If we're talking about a nucleoside, so it means that we're going to have no phosphate. While a nucleotide is going to contain phosphate. And remember that we can have 1, 2, or 3 phosphates. And when it comes to the nucleic acid itself, DNA is going to be a polymer of deoxyribose nucleotides, while RNA is going to be a polymer of ribose nucleotides. Now we are going to proceed to look at the primary structure of nucleic acids. As you can see here, this represents a molecule. So this is just a segment of RNA. So why do I know that this is RNA? Well, there's 2 keys that are going to tell us that this is RNA. Key number 1, when we look at the sugar motif, you can see that we have specifically a ribose sugar because on carbon 2, there is the presence of a hydroxyl group. The other key of determining that this is a molecule of RNA is the presence of uracil. Okay? Remember, each nucleic acid has its own unique sequence of bases, and that's what carries the genetic information from one cell to the next. When it comes to the structure of nucleic acids, understand that every time we are linking 2 nucleotides to form DNA or RNA, we are going to be forming the same type of bond, which is called a 3' to 5' phosphodiester bond. And this 3' to 5' phosphodiester bond comes from the idea that the hydroxyl group, that it is in the 3' position in your sugar motif. And right here, this is illustrating what will happen in DNA. And again, I know that it is DNA because my carbon 2 is a CH2. So carbon 3, which has a hydroxyl, is going to be bonded to the phosphate in the incoming nucleotide to make a 3' to 5' phosphodiester bond. I should note that when this chemistry happens, you do not do it on a monophosphate. The incoming nucleotide must be a triphosphate nucleotide. Here we're just illustrating a monophosphate just to give you a smaller picture, but understand that a triphosphate is the one that binds. Now, when it comes to the sequence itself in this primary structure for a nucleic acid, understand that we are going to be writing it from the 5' phosphate to the free 3' hydroxyl group, okay? So as you can see, this is the 5' end, and this is the 3' end. And To symbolize what are the sequences present, we just use the 1-letter code for the nitrogen bases found in the nucleic acid. As you can see, here, this is representing this small segment of RNA that we have in the image on the right side of the slide. The A, I'm just going to highlight them so you can follow. The A that we have here represents the adenine. The C represents the cytosine. The G represents the guanine. And the U is representing the uracil in the structure. Understand that each one of these nitrogen bases are linked to a sugar motif. And these sugar motifs are bonded to one another through that 3' 5' phosphodiester bond. So as you can see, we have several of those in the structure. Now we are going to shift our focus to specifically look into the structure of DNA, okay? Understand that the structure of DNA is actually a double helix. So there are 2 molecules, 2 strands of this nucleic acid in order to make the molecule of DNA. So the molecule of DNA, these 2 strands, are actually held together through hydrogen bonding events between the nitrogen bases that are present on each strand. So during the 1940s, biologists determined that the bases in DNA had a specific relationship. The amount of adenine is always going to be equal to the amount of thymine, and the amount of guanine is equal to the amount of to the amount of cytosine. The reason is because scientists subsequently determined that the pairing, okay, of these nitrogen bases on each strand goes in the following manner. As you can see, adenine will always be paired with thymine in the structure of DNA, and guanine is going to be paired with cytosine in the structure of DNA. So whenever we have these nitrogen bases interact with one another, understand that that's what we understand it as complementary base pairs. And when it comes to DNA, these complementary base pairs are going to determine that we have in these structures equal amounts of A's and T's and equal amounts of C's and G's. That means because they're base pairing with each other. As you can see, the interactions that are holding together these base pairings are going to be hydrogen bonding events. And they are represented by those dotted lines. I'm going to highlight those dotted lines in blue. So this is the hydrogen bonding events that are happening between adenine and thymine on the left. And we have the hydrogen bonding events for guanine and cytosine on the right. Notice that I am not requiring you guys to know specifically which atoms are undergoing hydrogen bonding. You just need to know the number of hydrogen bonding events. So in the case of adenine and thymine, we're going to have 2 hydrogen bonding events, while in guanine and cytosine, we have 3 hydrogen bonding events. Now, when it comes to the structure of DNA, understand that we're going to have 2 strands, okay, that are going to be held together by the hydrogen bonds of those nitrogen bases, okay, that are going to happen between adenine and thymine and guanine and cytosine. So in this picture, we are illustrating specifically those hydrogen bonding events that are occurring between the nitrogen bases. Notice that the strands in the double helix in DNA are going to be anti-parallel. Which means that one strand runs in the 5' to 3' direction, while the complement of it runs in the opposite direction, 3' to 5'. And you can see it illustrated by these symbols that are specifically in that lavender ribbon that we have in the structure, okay? Now, understand that when it comes to the structure of DNA, Watson and Crick were the ones that were awarded the Nobel Prize for determining the structure of DNA. But in reality, understand that there was another scientist named Rosalind Franklin that also made major contributions to understand the structure of DNA. So this is the reality. In 1953, James Watson and Francis Crick proposed that DNA was a double helix. They also suggested that it consisted of two polypeptide strands, and again, a polynucleotide strand, that they are winding around each other like a spiral staircase. And remember that these two are going to be anti-parallel. One runs 5 to 3 and the other one 3 to 5. Then they also demonstrated that they contain sugar phosphate backbones that are analogous to the outside stair railings, and then the bases are arranged like steps along the inside of these strands. As I mentioned before, they also determined that they run anti-parallel. One runs 5 to 3 and the other one 3 to 5. The reality is that Watson and Crick were able to do all of this work because Rosalind Franklin at that point was a grad student, and she actually took an X-ray image of the structure of DNA. At that time, what happened was that because she was a grad student, her professor that she was working under sent that image to Watson and Crick, so they were able to make their predictions on the structure of DNA. And if it wasn't for the image that she made or she obtained through her experiments, they were not able to make these predictions. Understand that the reason why she was not awarded the Nobel Prize is because she died before the Nobel Prize was awarded, and Nobel Prizes are never awarded to people that are deceased. So understand that when it comes to the structure of DNA, the contributions of all these three scientists were the ones that were able to make these predictions and these discoveries available. And it is currently what we know, what is the structure of DNA.